So students, I have been saying it again and again that regulation of a metabolic pathway is as important as the metabolic pathway itself, or perhaps it's more important. And the PPP is not an exception. We have seen that the pentose phosphate pathway have two divisions. One is the oxidative phase, and the other is in non-oxidative phase. Similarly, we can also say that one is the phase which leads to the formation of a very important compound, glucose 6-phosphate, and this glucose 6-phosphate then is involved in many important reactions, and also we get the NADPH during this phase. And then there is a biosynthetic phase in which you get different substances which they connect the pentose phosphate pathway with the glycolytic pathway. Now the regulation is very important because in certain circumstances you really need NADPH, and we will see that what are those circumstances. And then in certain circumstances you want to enhance the glycolytic pathway. So in different tissues the demands are different, so a metabolic pathway have to be modulated or regulated in accordance with the demand of the tissues. And this is what regulation is all about. Let's see it in a little bit more detail. As I have already told you that the first reaction of PPP pathway is very important because it is the rate limiting effect. It is a rate limiting step rather. Why is rate limiting step? Because it is acted upon by enzyme known as glucose 6-phosphate dehydrogenase, and this enzyme is involved in the regulation of this whole pathway. This enzyme is the most important perhaps component as far as the regulation of this pathway is concerned. This enzyme doesn't have only catalytic activity, of course, because of its catalytic activity, it gives rise to the formation of gluconate compound, but also you get NADPH because of its action, and this NADPH then modulate the activity of this enzyme. NADPH activate or inhibit this enzyme through the feedback mechanism. For instance, of course, you know about feedback mechanism. If the product is enhanced in amount more than what the body requires, it will then revert the reaction in a backward direction by negatively regulating the reaction by its action on the enzyme. तो क्या होगा जब NADPH ज़्यादा पैदा हो रहा होगा तो it will regulate the glucose 6-phosphate dehydrogenase in a negative way. It will inhibit the reaction. Higher NADPH, inhibition of the reaction. Lower NADPH, reaction will be enhanced, the pathway will be enhanced. You can also say that if your body, your cellular compartments have lesser amount of NADPH, what will happen that there will be a tendency for the reaction the pathway to get enhanced because the NADPH is needed now. And when there is no need for NADPH, there is lesser demand of NADPH, the pathway, the PPP pathway will be inhibited or slowed down. And this all takes place because of the modulation of because of the regulation of the enzyme, which we have already said you. The enzyme which is modulated here is glucose 6-phosphate dehydrogenase. But it is not always the feedback mechanism which really regulate a reaction, a pathway. Also, sometimes is of great importance is the feed forward way of regulation. What is feed forward mechanism? Is that if a product of a reaction control and enhance the reaction which are occurring downstream of that reaction, I mean downstream of the part where product is being formed. So, if a reaction is giving rise to a product B through a product A, the product B then can enhance a product C which is being formed in the succeeding reactions. So, that is the feed forward mechanism. So, here what will be the feed forward mechanism in case of a PPP? The feed forward, for one example of feed forward mechanism is here is the formation of ribulose 5-phosphate by the activity of or by the conversion of what we say the glucose, the gluconate phosphate. The gluconate phosphates under the action of glucose 6-phosphate dehydrogenase gives rise to ribulose 5-phosphate, and then ribulose 5-phosphate is then we can say is the glucose 6-phosphate dehydrogenase in this way positively regulate the formation of ribulose 5-phosphate which is occurring ahead of the formation of glucose, ahead of the formation of the product which is controlled by glucose 6-phosphate dehydrogenase. That is the example of feed forward reaction. So, we have seen here that it is the regulation of PPP is a combination of different enzymes, a combination of the need of the cell to cope with its demands, to fulfill its demands. If NADPH is lesser, then the reaction will move in the forward direction. If less NADPH is in a much higher amount, the reaction will halt it. Similarly, glucose 6-phosphate dehydrogenase will act in a way that it enhances the production of, let's say, for instance, ribulose 5-phosphate or ribose 5-phosphate by feed forward mechanism. And in this way, because of this combination of mechanisms of the regulation of the pathway, the overall homeostasis of the cell is maintained.